Hi everyone, my name is Corey Padilla and I'm here from Dovetail Genomics to talk to you about how you can use our new HiChIP kit to investigate protein-directed chromatin architecture. To start, we'd like to orientate you to the workflow of our HiChIP kit. It starts with cross-linked cells with an input of down to 1 million cells. From there, we fragment the chromatin using MNase, which eliminates any need for sonication and therefore streamlines the workflow quite a bit. After that, we do our immunoprecipitation with either a set of validated antibodies or we provide a protocol for you to validate your own antibody of choice. From there, it follows a very standard proximity ligation method where free ends are ligated, then the crosslinks are reversed, and then we generate a sequence library. The resulting ChIP-seq libraries contain similar characteristics to that of ChIP-seq data. Now, to demonstrate this, we have a coverage profile of CTCF ChIP-seq data on the top in blue, and below that, we have CTCF HiChIP data in green. And as you can see, the peaks between these two coverage profiles align quite nicely. There's an added benefit, though, of the HiChIP of being able to look at the conformation signals that occur between these peaks, and that's indicated in these contact arcs that are drawn between the peaks that are indicative of the contact frequency between these regions of interest. Now, if you were to zoom in on any one of these peaks, you can see that we can phase nucleosomes, their bottom in the green profile, where you can't with ChIP-seq data because we use MNase to fragment the genome. Now, there's an added benefit of using a protein-directed approach to capture conformation events in that it increases your signal-to-noise ratio while decreasing your sequence cost. So what we have shown here are two contact matrices that are metagene analyses around high occupancy CTCF sites for HiChIP on the left at 150 million read pairs, and on the right with a restriction enzyme approach at 800 million read pairs. And as you can see at the CTCF site, there is a very bright spot on the contact map for the HiChIP data on the left with very little noise around it, whereas with the Hi-C contact map, you do see an increase in brightness around the CTCF, but it's associated with a lot of noise. And if you were to calculate with the signal-to-noise ratio based on coverage at target sites versus away from those target sites, you can see that the HiChIP data has a signal-to-noise ratio of 2.5, where the multi-restriction enzyme approach that we're displaying here only has a signal-to-noise ratio of less than 1. And so to end, I'd like to show the current suite of validated antibodies that we have in the left column. And on the right, these are antibodies that we're working to validate. And again, if you don't see the antibody that you're interested in using, that's fine. We have a protocol that will allow you to validate your antibody of choice within the Dovetail HiChIP kit. So with that, I want to say thank you for listening. And if you have any questions, please feel free to reach out.